So, in today's video, we're going to cover 5 must-known concepts to physiology, which hopefully is going to make your physiology learning a lot easier and more fun. So, the first concept is body fluid. In the average 70 kilogram man, 60% of the body weight equates to 42 liters of the body fluid. And of the total body fluid, we can separate it into ICF and ECF. ICF is the intracellular fluid, and ECF is the extracellular fluid. So 1/3 of the body fluid goes to the ECF. And from the ECF, 1/4 goes to the intravascular fluid, basically the plasma inside your vessels, and 3/4 goes to the extravascular fluid. So right now, we're just gonna pump in the rough numbers. So this is around 28 liters, 14 liters, 3 liters, and 11 liters. This is not the exact number, but just bear with me. And from the extravascular fluid, we can also separate it into the interstitial fluid and the transcellular fluid, which if we were to pump in the numbers, will be about 10.5 liters and 0.5 liters. So that covers the first concept of body fluid. As for the second concept, which is going to be ECF, ICF, and its relation to sodium and potassium. So you have to remember, sodium is always high in the ECF, extracellular fluid, whereas potassium is high in the ICF, the intracellular fluid. And chlorine always follows sodium, so it's also going to be high in the ECF and low in the ICF. Okay, and one way to remember this is by thinking outside salty and inside crazy. So salty being sodium, so outside salty, and crazy, you can think of the K as crazy, and K is inside, so inside crazy. And the importance of this relation is because we want to maintain a negative resting memory potential, which is to say the ICF is relatively negative compared to the ECF. And this is important in nerve impulse transmission as well as muscle contraction. So the third concept covers the cellular transport, under which is active transport and passive transport. Now we're just going to breeze through it and mention some clinical scenarios as well as important facts. So active transport requires the usage of ATP and is against the concentration gradient, whereas passive transport does not require the usage of ATP and it is along the concentration gradient. So for facilitated diffusion, you have to know the purpose. Why do we need facilitated diffusion? It's because the lipid bilayer, it cannot transport large and polar molecules. So that's why we have proteins that aid in the transport of large and polar molecules. And then there's the simple diffusion. So just know that simple diffusion can occur in the living as well as the non-living. And then there's the ultrafiltration, which actually happens in the kidney, at least one of the location. And then there's the osmosis. And here we have 3 clinical scenarios. So let's say there's a hypertonic chyme. The result of hypertonic chyme is diarrhea. And for osmotically active lactate, what's going to happen is lactose intolerance. And finally, in osmotically active glucose, we're going to have polyuria. So that covers the third concept in physiology. Fourth concept is cell communication. So for cell communication, there's a few different categories, but we're going to cover the first one, which is local and long distance. So local is basically between gap junction or autocrine and paracrine. So autocrine, you can think of it as the cell produces signaling molecules and it uses it. Whereas paracrine is the cell produces the signaling molecule, but it acts on other cells. And one example of autocrine would be interleukin 1, which is produced by macrophage and monocyte. And for paracrine would be histamine, for example. And for long distance, we have endocrine and neurotransmitter. Neurotransmitter doesn't just act in the synaptic cleft, it can also act elsewhere. So in this case, we call them neurohormone. And beyond that, for cell communication, we also have the electrical and chemical. And electrical, we're going to talk about resting membrane potential. So this is the fifth concept, RMP, or resting membrane potential. So think of resting membrane potential as the baseline electrical charge across a cell membrane when the cell is at rest. It's like the cell's default setting. Now, in most cells, the resting potential is negatively charged inside compared to the outside. And this relates to the previous concept of nerve impulse transmission as well as muscle contraction. So how do you maintain the resting membrane potential where the inside is more negative? There's three ways. The first way is the sodium-potassium pump. So the sodium-potassium pump basically pumps 2 potassium into the ICF and 3 sodium into the ECF. And what this results in is a negative charge on the inside. And beyond that, we have negative protein, the ICF, which maintains the negative RMP. And finally, we have the high potassium concentration in the ICF, which leaks to the ECF. And this also results in less positive charge in the ICF. So to elaborate further on the RMP, we're going to talk about local potential. So local potential is a general term. And under local potential, we have graded potential and actual potential. So now that you've heard of the term of RMP, graded potential, and actual potential, we can actually combine everything and fit them into a timeline. So we begin with the RMP. So the resting membrane potential is supposed to be negative, but say it increases, it becomes more positive. When it becomes more positive, it produces a graded potential. And once it produces a graded potential, it can actually go on and propagate as actual potential. And finally, it reaches the synaptic activity. It could be neuromuscular junction or even connection with other dendrites and axons. So to elaborate further on the graded potential and actual potential, we have to know a few differences. For graded potential is usually short distance, and actual potential is long distance. And for the graded potential, the higher the stimulus actually produces higher potential strength. The potential is actually stronger. Whereas in actual potential, it's always the same amplitude, regardless of how strong the initial stimulus is. So let's elaborate further on the synaptic activity. So again, it's just going to be a repeat. It's actually a cycle. So the actual potential arrives at the presynaptic, and it causes the neurotransmitter to be released and goes across the synaptic cleft and binds to receptors on the postsynaptic membrane, and this eventually produces more actual potential. Now, to elaborate further on the neurotransmitter release, so neurotransmitter, there's two types in general. There's the ones that produces the EPSP, and there's one that produces IPSP. EPSP is excitatory, hence E, whereas IPSP is inhibitory. Okay, so under EPSP, there's acetylcholine and glutamine. These are neurotransmitter that produces potential, whereas IPSP are glycine and GABA. So glycine and GABA are ones that produce inhibitory potential. And so if we have the IPSP, inhibitory postsynaptic potential, it's going to make it hard to generate actual potential, and thus hard to generate muscle contraction. And if there's one more thing I'd like to talk about, it would be the all or none law. So for actual potential, it's simple term. It means that once a neuron gets a strong enough stimulus to reach its threshold, it fires an actual potential at full strength. And if the stimulus isn't strong enough to reach the threshold, nothing happens. There is no partial action potential. It's an all or nothing deal. So this is all or none law. And that concludes today's video.